Tyrrell. I'm an assistant professor at the University of Alabama at Birmingham, and I'm here with Dr. Arvind Pandey, who is the Werner Risau Award winner in vascular biology.
Thank you.
So you have some interesting work on endothelial cells and CD70, and I think the first thing that I'm kind of curious about is how did you conceive of this project and where did this all start?
Yeah, no, it's been an interesting project for sure. It was something that actually grew out of some work I had done at Vanderbilt when I was there for my postdoc, and came over here to Brigham to do my vascular medicine fellowship as well as junior faculty time. And so it was a project that started off there and just kind of continued with me when I came over here, and utilized a lot of the tools and a lot of the new techniques that I had access to here to kind of build that project forward.
That's interesting that you were able to bring it from postdoc time to another institution, kind of building on both worlds, I guess. But from that, was there any difficulty in doing that or anything? I guess you mentioned the new technologies and things that were available here that you could do. So can you tell us a little bit more about what new types of avenues you were able to take when you got here?
Yeah, it's always fun to be able to take a project and then be able to add in new pieces as you get the opportunity to do more things. One of the really important pieces of technology that we ended up using in the paper was the use of genetically encoded biosensors, and that's something that one of my co-mentors for the project, Dr. Thomas Michel, has been a really great expert and world leader in fact, for utilizing those biosensors. So it was a really great opportunity to be able to learn more about their use in biology as well as utilize that for my project. And then my other primary mentor, Dr. Lescalzo, has had a long history of doing a lot of great work with nitric oxide and reactive oxygen species, so bringing that part into the mix of then understanding how CD70 influences those parts of endothelial biology was really, really fun.
Yeah, that's really interesting. The biosensors especially are really a really neat technique that is not widely available, so that's definitely interesting. Can you tell us a little bit about what might have been a hurdle that you had to overcome or something difficult, like either publishing the paper or carrying out the research?
I think anytime, like you alluded to before, anytime you're changing institutions, it creates roadblocks and speed bumps. So certainly, and I was going from being a postdoc to going back into doing clinical fellowship time and then coming back into the lab. And so that stop and start was a little bit tough to keep all of the momentum going. But I think that's where the guidance of the mentors really is very helpful. And so credit to Dr. Michel and Dr. Lescalzo for really being very, very willing to kind of accommodate my several different responsibilities I was trying to juggle as I was getting the project forward.
And is it difficult now that you're doing clinical work to balance the research side and the clinical side?
Yeah, I would say that's the challenge I'm living through of the physician-scientist world. I think, like everyone, grant funding and all the things that it takes to try to do good science is an ever-present issue. But then balancing in seeing patients, and I enjoy clinical work, and so it's hard to say that I don't want to do that. And finding the right balance of being able to do that as well as the basic science is tough. But I will say that one of the exciting parts of doing the clinical work is that it can provide inspiration for some of the basic science questions I'm interested in asking. And so that part is one of the benefits of trying to balance the two.
Yeah, that's interesting. I have a more scientific question. So you, in the paper, in the introduction, I think you mentioned that CD70 is typically known for roles in antigen presentation, but endothelial cells have been shown to play some roles in antigen presentation. So do you think CD70 plays a role in the endothelial antigen presentation?
Yeah, no, that's a great question, and I think something we don't fully know yet. It wasn't something that I explored too much in the initial set of studies, but I think it's a very good question, and certainly something that we're interested in exploring further. I think like you mentioned, the antigen presentation part of endothelial cells is still something that we're understanding and trying to figure out what are the contexts when that happens, and and what function that serves. And so how CD70 interfaces into that function of endothelial cells is certainly a question we don't fully know the answer to yet.
It seems like you have a lot of interesting avenues to take with CD70 because it's only recently been shown to have a pretty big role in endothelial cells specifically. Can you tell us a little bit about what types of things or questions you might be asking in the future?
Yeah, I think the big thing I'm interested in from the endothelial standpoint is there are a lot of observations we've made in this initial paper, but I think still trying to understand mechanistically how is CD70, how is the signal going from CD70 to the, eNOS and nitric oxide? What are the mediators of that, and how can we potentially influence that from a therapeutic standpoint? Are questions that are things that we're looking into now. I think the other part of this that I mentioned towards the end of the paper, but is an active area that I'm trying to flesh out, is how does CD70 interact with other members of the TNF superfamily? I think that part is very unexplored. In TNF superfamily members in general, it seems like a lot of them function is described in isolation, but how they might all interrelate, I think, is a question that is not fully understood. And so how does CD70 interact with them in the context of the endothelial cell is also a question I'm very intrigued by.
Yeah, that's really interesting. So I'm curious if you wanted to explore the role, that interconnection. I think a lot of research, especially in inflammation, gets kind of washed out by TNF alpha.
Yeah.
But are there other TNF superfamily areas that you're interested in looking?
Yeah, there's a few that have been evaluated already. So OX40 ligand is one example that comes to mind that people have looked at in the context of endothelial cells, and how that might interrelate with CD70 is intriguing. I will also say that a lot of the classical description of CD70 is in the context of CD70 and its receptor CD27, but it's not known how CD27 might interplay in this, these observations we made of CD70 in endothelial cells. And so how does CD27 impact any of these observations is also unknown.
So I just want to ask, because you are practicing in the clinic, if this work that you've published in ATVB impacts your clinical practice or clinical observations.
Yeah, it certainly does. I think as we were talking about before, how the clinical work can influence the research. The research can also then influence what we may be able to utilize from a clinical therapeutic standpoint. And so I think the part that's really inspiring for me is, there's a lot of patients that I see that have really difficult to treat vascular reactivity issues, and we don't understand what causes them to experience that. A lot of times it interfaces with rheumatologic conditions, and our ability to treat those patients is very limited. And so a project like this that is exploring a completely new avenue for understanding endothelial function and, in consequence, potential implications for vascular activity is certainly something that I think helps me stay motivated to try to understand how does CD70 or TNF superfamily members in general impact endothelial function, so that we could potentially open a new avenue for therapeutics going forward.
Thanks. That's very interesting. And I just want to say congratulations again on this work, and thanks for being here.
Thank you.